All right, welcome back everyone. Thank you for joining us. We are moving on to our next presentation, which is on genetics of Pompe disease. And we are thrilled to have Kare Anstett here. Kare is a certified genetic counselor in the Department of Neurology at NYU Langone Health in New York City, where she provides genetic counseling services for the Comprehensive Neurofibromatosis Center, the Lysosomal Storage Disorder Program, and the Division of Neurogenetics. She is engaged in clinical research projects in neurofibromatosis and lysosomal storage disorders, presenting at national and international meetings. She is also a clinical supervisor, research advisor, and guest lecturer for the Joan H. Marks Graduate Program in Human Genetics at Sarah Lawrence College in Bronxville, New York. Kare, we're thrilled to have you. Feel free to take it away.
Thanks, Elise. So I'm going to give a presentation that gives an overview of different genetic aspects of Pompe disease, including just a general introduction about genetics and how it relates to lysosomal storage disorders, the role that genetics plays in our enzyme levels and how that causes the different types of Pompe, different testing options that are available for both diagnosis and carrier screening, the inheritance pattern of Pompe and how that affects recurrence risks within families, as well as options for family planning and family screening. So I like to think of genes as recipes to make all the parts of our body, and the genetic code is made of the 4 letters A, C, D, T, G. And with the recipe that each gene is, it provides instructions for different parts of our body. And in the case of most of the lysosomal storage disorders, it provides instructions for an enzyme, which job is to break down one substance into another substance. And if we take this analogy that our gene is like a recipe, a spelling mistake in the recipe can mean that the enzyme that's made is not made correctly and cannot function properly in the body. And in the case of Pompe disease, sorry, I'm having some trouble with advancing the slides. There we go. So there's different mutations within the gene for Pompe disease. Some mutations are a little bit more tolerated and the gene is still able to function to some degree. So you have a lower level of enzyme than is expected. And in other cases, a mutation might have a more detrimental effect on how that gene is read by the body. And so you end up with very low or no functional levels of enzyme. Now, everyone has 2 copies of these genes. And so someone with Pompe, we look at the 2 mutations that they have in combination. If those mutations are more likely to cause very low or no enzyme levels or some level of functioning enzyme, and those 2 in combination give your overall enzyme levels and can be an indicator of whether someone has early onset Pompe or late onset Pompe. This can be particularly important when we're talking about family planning or the risks to future children if 2 people are carriers for Pompe, or if someone is identified on something such as newborn screening and we don't yet have symptoms. And so I have this graph that kind of illustrates the different enzyme levels that we can see. On the far right-hand side, we have our full functioning levels of the GAA enzyme. And then if we move towards the left, as the purple color gets lighter and lighter, you have your no functional enzyme. And so everything exists on a gradient. When we look at the full enzyme levels, we have people that are non-carriers for Pompe, so they don't have any mutations in either copy of their gene. And then as we move leftward, we have people that are carriers. They have a single Pompe mutation. And as you can see on this diagram, there is an overlap between carriers and non-carriers. So enzyme testing is not a good way to check if someone's a carrier because they could have the same enzyme levels as someone who is a non-carrier. Then as we move farther to the left, we hit a threshold where the enzyme level gets to a point where it's too low to do the functioning job it has in the body, and then we have symptoms developing. We have those that have about 2 to 40% of the normal expected enzyme levels, and they have the late onset form of Pompe. And then we have, on the lower end, those with infantile onset Pompe that have less than 1% usually functioning enzyme levels. And so that's how the two mutations that come together can lead to an overall enzyme level that predicts whether someone has infantile or late onset Pompe. And there's different testing that we can do for diagnosis and to find carriers of Pompe. And this table gives an overview. So we can do enzyme activity level, which, as I said, is good for detecting people that have Pompe, but is not effective at detecting carriers. There is a limitation called pseudodeficiency for enzyme testing, which I will explain in a moment. We can also do genetic testing for Pompe, and there's two main types. There's targeted variant analysis, and then there's also full gene sequencing, and I'm going to explain the differences between the two of them. Both of those genetic tests can be used to detect people that are diagnosed with Pompe, and they can also be used to detect carriers. There are a couple limitations to both of those testing options as well. And then I'm sure it was discussed a couple times in the other presentations, but there are other markers of Pompe that we can see in people that are affected. For example, the creatine kinase levels that we see in people that have excess muscle breakdown, as well as the urine hex4 levels. The thing about creatine kinase and urine hex4 is those can be elevated in people with other genetic conditions as well. And so they're not diagnostic of Pompe, but they might be done as part of a workup for muscle disease, and then can also be used to monitor Pompe over time. We don't use creatine kinase or hex4 to screen for carriers. So what is pseudodeficiency? The thing about laboratory testing for enzyme levels is what they use is they use a substrate that's supposed to mimic the substrate that naturally occurs in our body that the Pompe enzyme is supposed to break down. The thing about this laboratory created substrate, it's not exactly the same as the one that we create in our body. And so when someone has pseudodeficiency, we run the laboratory test to check enzyme levels, and it comes back and says, the enzyme's not functioning correctly, it's not breaking down this laboratory substrate. But if we were actually able to look inside that person's body, we see that their enzyme is actually able to break down the naturally occurring substrate just fine without any issue. And so what leads to pseudodeficiency is there are people that have genetic variations in their Pompe gene that affects how the enzyme reacts to the laboratory created substrate, but doesn't affect how it works in the body. And sometimes with genetic testing, we can actually find these variants that cause pseudodeficiency in order to better understand when we get low enzyme results back, but maybe someone is not presenting with symptoms as we would expect. And then, so this is a slide to explain the difference between targeted analysis and full gene sequencing for Pompe disease. So going back to our analogy that a gene is like a recipe or a very long sentence, when we do genetic testing, we're essentially running spell check on the genetic sequence to look for any changes from what we expect it to look like. So if we had the sentence, the gray cat ran down the hall, and we were going to do targeted analysis on it, we would just check a couple spots along the gene, maybe places where variants or mutations are more commonly seen, maybe depending on someone's ethnic background where they're more commonly seen, but we wouldn't check the entire gene. So this type of testing might be available on carrier screening and might be used to reduce the risk that someone is a carrier, but it's not checking the entire gene as we would do if we were doing full gene sequencing, where we're going from the very beginning to the very end, and we're able to assess for those rare variants. So if, for example, someone had a mutation at this spot, the C in cat for this sentence, it would be missed on the targeted variant analysis, but would be detected on the full gene sequencing. The other difficult thing about full gene sequencing, though, because we're looking at all spots along the gene, is sometimes we see something rare that hasn't been seen before, and we have difficulty understanding if it leads to low enzyme levels and leads to Pompe, or if it's one of those more benign variations that doesn't cause problems. which leads to my next slide that talks about variant classification. So for anyone who's had genetic testing done in themselves or a family member, there's different terminology that's used. We use the word variant usually in place of mutation. And variant just means that it's a change in what we would expect to see in that spot in the genetic sequence based off of sequencing that's been done in other people. So we have databases of various populations where this is what we expect the genetic sequence to look like, and if it looks different than that, then we call that a variation or a variant. And then we classify those variants based off of what we expect its role in is in developing human disease. So we have pathogenic variants, and that means it's a variant that we know leads to development of symptoms and a medical condition, either because we've seen that variant in other people before, or because some sort of characteristic of the gene change, we know that it's not tolerated and it doesn't function correctly. On the other end, we have benign variants, which includes pseudodeficiency variants, where it's a change in the expected sequence, but we know it doesn't lead to symptoms. And then right in the middle, we have something called uncertain variants. And those are variants where usually they haven't been seen in very many people or any other people at all. And we're not sure because we don't have enough evidence to say, does it lead to symptoms of disease or does it not? We also have a couple other categories in between these three. So you can have likely pathogenic, where we're a little bit more confident it leads to symptoms, but not completely sure. And then you can have likely benign, where we're pretty sure it doesn't lead to symptoms, but we need a little bit more evidence. And then we also have terms that we use to describe the characteristics of each variant. So this is going back to that other example with the sentence. And different changes or mutations we classify in different ways. So we use the word missense to imply that one letter in the genetic code is switched to another letter. We have insertions to show that different letters are added in, and deletions to indicate that other letters are deleted from the gene. And then we can have duplications where the same letters are repeated again in the genetic sequence. We also have something that we call nonsense variants. And I like to think of these like our gene sentence has a period at the end to tell the cell, this is the end of the sentence, the gene is finished, go and make it into a protein or an enzyme. And a nonsense variant puts that period too soon along the sentence. And then what's made is too small and is not functioning correctly and usually gets broken down. So in Pompe, for example, the nonsense variants, because they have such a great effect on the structure of that gene, those are more likely to lead to very low or no enzyme levels and be associated with more of the early onset forms. And to speak a little bit about newborn screening for Pompe, which is a type of testing that's available. There are different states that offer newborn screening universally for Pompe. This is the most recent as of this month. So all of the states that are blue offer Pompe disease screening using various methods. And the ones in red do not have it on their universal screening panels yet, but might be doing pilot programs or testing out that method. And to speak a little bit more about how newborn screening works, the purpose of newborn screening for various genetic conditions is to cast as wide a net as possible to capture as many possible infants that might be affected with a particular condition. The thing about casting such a wide net to reduce the chance of a false negative for newborn screening is that it increases the chance of false positives because it captures extra people by casting that wide net. And so anyone who screens positive on newborn screening does not necessarily mean that they have a diagnosis of that condition. It means that additional testing is needed. Usually the structure of newborn screening for Pompe starts with analysis on the dried blood spot that's given as part of the screening, and they check the enzyme levels for Pompe. If the enzyme levels are normal, that's a screen negative. If the enzyme levels are low, then that's a screen positive. And in some newborn screening programs, as soon as the enzyme level is low, they contact the family and the pediatrician, and the infant is referred to a newborn screen follow-up clinic for diagnostic testing and further evaluation. In other programs, they do an extra step before they contact the family or their provider, and they might do genetic testing on the dried blood spot. If they find variants in the GAA gene, then they will contact the family for that follow-up testing and that referral to the newborn screen clinic. If there aren't any variants detected, then that would become a screen negative result. To speak a little bit more about the inheritance pattern of Pompe and recurrence, I have a couple diagrams to illustrate. So as many of you might be familiar with, Pompe is a recessive condition. So you are affected with Pompe if both copies of your gene from mom and from dad carry mutations. So once someone is diagnosed in the family with Pompe, we look to the risk of brothers and sisters, either those that are already born or future children that are being planned by the parents. And the following risks apply with natural conception. So there's a 25% risk that a sibling will be affected with Pompe. There's a 50% risk that they will be a carrier, much like their parents are, which can have important implications for future family planning when they are older. And there's a 25% chance that they're unaffected and they don't carry any mutations at all. Now, in families that have a child that's diagnosed with Pompe, sometimes the perceived risks might change a little bit depending on the type of Pompe diagnosis. So for example, if the diagnosis is for infantile onset Pompe and there are brothers and sisters that are much older, their risk is probably less than 25% because they're older than the expected age that symptoms will start. But in a family with late onset Pompe, there can be a wide range of when those symptoms can start. So the risk is much closer to that 25% of being affected. And then moving on to adults with late onset Pompe and thinking of their own family planning for future children, the risk to children is really dependent on the genetic status of their partner. And so in most cases, a partner of someone with Pompe is a non-carrier and is unaffected. And so every child that's born from that couple will be a carrier for Pompe because they will always inherit a mutation from their one parent that's affected. But in some rare cases, the other partner can also be a carrier. And the chance of this happening varies a little bit depending on someone's ancestry, but is usually less than 2% or 1 in 50 chance of that happening. But if the partner is a carrier, then the risk changes a little bit. So 50% of children are at risk of being carriers, and 50% of children are at risk of being affected with Pompe, with natural conception. And the type of Pompe that those children are at risk of changes depending on the specific genetic mutations that are in the parents. So depending on the mutations that are present, it is possible that children of someone with late onset Pompe might be at risk of infantile onset Pompe. So it is really important to discuss this with a genetic counselor because that can have important implications for which family planning options someone is considering. So to move on to different screening and family planning options that are available, I like to kind of categorize these into two general areas. So one is the testing options and planning options available if we have genetic testing done in the person with Pompe and we know their mutations. And then the other is testing that does not involve knowing the mutations of the person with Pompe. So when we know the mutations, we can do preimplantation genetic testing, or PGT, which is done prior to conception. And then we can also do prenatal diagnostic testing during pregnancy, and I'm going to explain those two options in detail in a moment. We can also do genetic screening of family members that are at risk of being carriers, as well as family members that are at risk of developing Pompe by testing them for the specific variants that have already been identified in the affected family member. Other options that don't require the genetic mutations include enzyme testing for family members that are at risk of symptoms of Pompe, which, as I said previously, is not effective for testing for carrier status. We can also do clinical monitoring for symptoms of Pompe. Parents can consider natural conception without doing any additional screening, or considering screening after a child is born. And some people might consider adoption for various reasons. So to speak a little bit more about those two family planning options in particular, so preimplantation genetic testing, or PGT, is a procedure that occurs in the context of in vitro fertilization, or IVF, done with a fertility clinic. And it's usually done by collecting the egg and the sperm from each parent and then injecting sperm directly into the egg for fertilization. Once the developing embryo is a couple days old, they will do a biopsy to remove a couple of the cells for testing. And then they'll do genetic testing on those cells to look for the mutations for Pompe that were present in the parents. And you will get results back that either indicate no mutations for Pompe, a single mutation indicating they're a carrier, or 2 mutations indicating the embryo is affected. And then you choose the embryos that are not affected with Pompe, so either no mutations or a carrier, for transfer to the womb for pregnancy. The thing about this type of testing and the IVF process, it is not always covered by insurance. There's a lot of variability from state to state, as well as from different policies about what types of procedures are covered. Sometimes part of the IVF process is covered by insurance and other parts are not. Or sometimes there might only be coverage for IVF for other reasons, for example, infertility, but not for coverage for the genetic testing or for the genetic risk. So it's something to really investigate if it's something that you're considering. Usually a fertility clinic can help you walk through all of your different options and what's covered and what's not. The other option for diagnostic testing for the risk of future children is prenatal diagnostic testing done during pregnancy. There's 2 time points where this might be done. Chorionic villus sampling, which takes a sample of the placenta, is done at the end of the 1st trimester. And amniocentesis, which takes a sample of the amniotic fluid from around the fetus, is done around 16 to 20 weeks gestation. Both of these samples, the placenta and the amniotic fluid, contain the same DNA as the fetus, and so is representative of their genetic makeup. But these are also invasive procedures that do carry a risk of complications that can include miscarriage. Many parents will consider this option after weighing the risks of the procedure if they're thinking about changing their pregnancy management decisions based upon it, or to prepare in advance for the birth of a child that might be affected. This procedure is usually covered by insurance, including the testing. And then I just want to speak briefly about pregnancy for someone who has Pompe. So women with late onset Pompe might be at risk of changes in their symptoms or progression of their symptoms during pregnancy. And these changes in their symptoms might increase the risk of complications. So for example, if someone has weakness in their diaphragm muscles that's affecting their breathing, it could affect their oxygen levels to both mom and to the fetus, which could have effects on the health of the pregnancy. And so it's really important that any women that have Pompe speak with their specialist prior to becoming pregnant to discuss their specific risks, depending on their disease status and their symptom status, and make a plan for their care during that time, which could include increased monitoring or increased testing during that period to ensure that they're healthy. So I thank you for your attention, and I'll now open the floor to questions.
Wonderful. Thank you so much, Kara. Yeah, we have some time to take some questions. We do have some that have come in already, so we'll start there. But please, if you do have some questions for Kara, please go ahead and type those in the Q&A box. The first question, Kara, I see is regarding suspected LOPD babies identified by newborn screening. How do we determine if a variant of unknown significance is potentially a pseudodeficiency allele?
So that's really challenging, especially when we're looking at late onset Pompe, because other types of tests that are available to assess if someone is symptomatic might not be apparent in someone who's very young. So checking creatine kinase levels or Hex4 in the urine. So the challenging thing with newborn screening is we're learning a lot of things that we weren't familiar with before, we weren't expecting. And so it can be really challenging, especially with these variants of unknown significance, determining what they mean over time. So I would say that what we have to do is we have to follow really closely and continue to follow up with any specialist that you've been referred to because of the newborn screening results to monitor that over time. And it's possible with more newborn screening and more genetic testing for Pompe that uncertain variant over time might be reclassified. And we could say that it's benign and a pseudodeficiency, or potentially that it causes Pompe.
Perfect, thank you. Another question, and I think you might have covered some of this when you went through kind of how things how these are passed down to generations. But the question is, our daughter was found to have LOPD on her newborn screening. Brother has been tested and is a carrier. Should we as parents do anything as carriers? Should we be tested? And what about our grandparents?
So good question. And I think this is the same attendee that says they weren't planning on future children.
Yeah.
So checking within the family for carrier status, the primary reason to check that would be for future family planning. So some parents might want to do the carrier screening, especially if their child has 2 mutations. So we know that one came from one side of the family and one from the other, because then more distant relatives, so for example, if these parents have brothers and sisters or nieces and nephews that are thinking about family planning, then those family members can choose to do carrier screening in preparation for future family planning. So it can be helpful to do that screening through parents and grandparents if it's to inform what carrier testing should be done for other people that are considering children.
Okay, perfect. Well, I think that sums up the questions that I see in the Q&A box right now. So thank you so much, Kare. I think that was a wonderful presentation.
Thank you.
Wonderful. All right. Well, thanks. Thank you, everyone, for joining us. We will be back at 3 o'clock, so in 9 minutes. And we have a couple exciting presentations to close out the day. We'll be hearing from Laura Case around PT and exercise in Pompe, Sareka Pendiali about nutritional considerations in Pompe. I will share a little bit of a mission spotlight, and my colleagues Brittany Hernandez and Mark Fisher are going to join us to talk a little bit about ways that you can get involved in grassroots advocacy. So we look forward to seeing you back here at the top of the hour at 3 p.m. Eastern in about 8 minutes. Thank you.